light. Okay, Igor, you can start your talk. Thank you very much.
Perfect. Thank you so much, and welcome everybody. Very nice meeting you, even if it's only virtually. As Fabio kindly introduced me, I'm Igor. I'm also from Italy originally, but I've been working with Roche for a number of years, and that gave me the chance to travel a bit around and to explore different parts of the world and different healthcare systems. And most of my time has been spent pretty much at the crossing between science, technology, data, and specifically in the area of diagnostics. So I'm going to start by giving you a very, very brief introduction about Roche and what we do. And then we're going to talk about something that is very close to my heart. I was telling the story early on to Fabio and Francesco about how I got my Russian name, but which is mostly due to my dad traveling back and forth between Italy and Russia. But there's also a secondary part of that story, which is also what brought me close to the oncology and the healthcare space, which is on the way back from one of his trips, he was quite close to Chernobyl when there was this nuclear accident. And so that's how we got sick with cancer. And I think that was, if you will, the seed for me to start to work in a healthcare space, that desire to understand complex diseases such as cancer. And so today, a lot of the things that we're going to talk about are going to be focused on the oncology space, not entirely, but they're also very transferable across different domains. So hopefully that will give you a bit of a, let's say, something to take home, even if you don't work in oncology. Okay, so let's start with the presentation. So, why is this not advancing? There we go. Okay, as I said, I want to give you a brief introduction about Roche. Many of you know Roche as a pharmaceutical company. And as a pharmaceutical company, sorry, I'm just going to need to get rid of this viewer here. Okay, as a pharmaceutical company, many of you know Roche has a lot of oncology drugs. But what many people don't realize is that Roche also has a diagnostic branch. And in this diagnostic branch, we make instruments and tests that are used in a variety of things. So when you go and send your blood test somewhere, chances are that it's going to be tested in one of those analyzers. And Roche is also a very large company with hundreds of thousands of employees, sites all over the world. And yeah, as I said, this gave me the chance to at least to experience the use of technology in so many different areas. And I'm going to try to focus today on specifically on the natural language processing part. But before I do that, I want to step back for a second and talk about the role of diagnostic in modern medicine. Pretty much everybody that has had any sort of health condition, or even just for a regular checkup, has sent some sample to a lab. And normally you get your blood taken, you send it out, and you get your result back in a day or two. And what happens is this gets sent to a centralized lab, gets put into one of those machines, and some data is generated, and associated with that data, there are some metadata. Now, this has been the standard of care for, let's say, many decades now, where a lot of these tests are very, very self-explanatory. You know, you have a glucose test, and you will have a range in which your glucose needs to stay. And based on that range, your physician can look at that report and say, great, you're not diabetic. Or they will look at your cholesterol, saying, maybe you should eat differently, and so forth. However, with the progress of technology, a new type of test has emerged, which is more complex test that cannot be interpreted simply and in a very, very straightforward manner. And so we're seeing the emergence of another set of skills and capabilities within the lab, which is what we call the dry lab, where the result of whatever came out from these machines get further analyzed and processed, sometimes using AI, sometimes using other type of algorithms. And it generates its own set of results that are a derivation of the first set of results. This is the case in complex diseases such as cancer, or in diseases that, let's say, require processing a large amount of data. Think, for example, if you're doing a prenatal screening for genetic disorder in a baby that is still in the womb of the mother. These are kind of complex tests that look at a lot of different data points and trying to get a result of something that is a very, very weak signal amongst, let's say, a very strong noise background. So I wanted to keep this in mind as part of this separation between wet lab and dry lab. And today we're going to talk a lot about what happened in the dry lab. But in order to do that, I need to take you back for a second into one particular part of the wet lab, and in particular in the field of genetic sequencing. So, and this is because this dry lab has taken really priority over three major areas of diagnostic procedure. Imaging, so when it comes to X-ray, MRI, CT, ultrasound, etc. All this data needs to be somewhat reviewed by human. And humans are very, very good at spotting certain things, but are very bad at certain other things. I think a good example is, you know, if you do an X-ray, a radiologist will hold up the X-ray against the light and look at the general state of, you know, your bones or your lungs, etc. But when it comes, for example, to oncology, if someone has a tumor, it's important to understand how big is that tumor, how many centimeters. And so accurately measuring that from an X-ray, it's something that, for example, machines tend to be much more accurate than humans. Similar thing happened with time series data. Think of electrocardiograms, echocardiograms, EKG, and so forth, but also your heartbeat signal, your glucose signal, etc. And then the third bucket is really this area that I was mentioning early on, that it's one of the theme of today, which is the omics. What do we mean by omics? It's genomics, proteomics, metabolomics, and a lot of these disciplines that look at very, very, in a very unbiased way, at a lot of multiple signals at the same time. So these data sets are very, very challenging for a human to interpret because of the sheer volume. And I'm going to show you in a second what I mean by that. And so it becomes very hard for someone to process that data in their mind. And so you need to rely on tools, informatic tools that help you to basically get a better sense of what is going on. Okay, now let's dive right into the clinical sequencing part. So you all know each and every one of our cells has a molecule of DNA inside the nucleus. There's the same copy of DNA in every cell. Some genes are turned on or turned off based on where they are, but basically the blueprint, what we call the blueprint of how your body functions, it's stored in each and every cell. And sequencing is the act of being able to read that sequence of letter A, C, G, and T, and basically record every single sequence of information and how it might be different from one individual to another. And these are numerous applications, but just as a quick detour and refresher of molecular biology for those that are not familiar with that space, your DNA code, it's very much like a binary code. Instead of having 2 bits, it has so it's basically, instead of having just that 1 and 0, it has 2 bits of information. There's actually 4 values, A, C, T, and G, and those values encode for amino acid. When there is a change, that might lead to a change in amino acid. What does it mean? Well, amino acids, once they are all strung together, they form proteins, and proteins are the main functional unit of what happens in your body. And so having a difference in the DNA might lead to a difference in the amino acid, and that might lead to the protein not functioning correctly, or just being different. We will see in a second that not all changes are necessarily bad. Sometimes changes are just natural variation. There are 4 different types of mutation. I'm not going to go and spend too much time into that, but basically there are mutations where basically a letter is swapped. There are mutations where a big chunk of code is missing. There are mutations where there's a almost like a copy and paste of like a duplicate of the same code repeated multiple times. Or there are mutations that we call fusion, where something that comes from one gene gets somehow pasted very close to something that comes from another gene and create a new molecule that shouldn't be there, quite frankly. So those are just for your reference. You don't have to remember it for that. The other part that is very important to understand, though, when it comes to mutation, it's the role of germline mutation versus somatic mutation. Now, when we talk about mutation, germline mutation, it's something that we carry from basically from our parents, from the sperm and the egg of our fathers and mothers. We carry those mutations, so they determine a lot of things about ourselves. How tall we will go, the color of our eyes. Some of them are complete normal and not not relevant from a health perspective. So, for example, the color of your eye, whether you have a fast metabolism or not. But some of them might lead to tell us that we have a certain predisposition for a disease. A very, very good example, it's the BRCA gene, which is related to a higher, a malfunction of the BRCA gene, or a faulty copy of that gene might lead to breast cancer. Or there are a number of conditions that are heredited in that way. The other type of mutation are what we call somatic mutation. And this is what happens often when you come to cancer, but also much more, let's say, innocent things such as birthmark or a mole or a benign tumor. Those mutations are localized, they're not carried into your offspring, and they basically affect a limited number of cells. They might be benign, they might be malignant. If they are malignant, we often refer them as cancer. Now, this is a very, very important thing, because we will see in a second, one of the key concepts that we need to remember when we go and do sequencing to extract information. So we go and read the sequence of letter. In the case of somatic mutation, we will actually have 2 separate sequences. We will have your germline sequence, which is what you have in all your cells, plus we will have a sequence that comes from the tumor, for example. And so this is what we call somatic mutations. And it is very important to distinguish between those 2 signals also when we come to the interpretation of these exams. Okay, with that in mind, how do we get DNA to do this analysis? Well, in oncology space, the most common way is to actually do a biopsy. So very often when it comes to a suspicion of a tumor, the patient will be taken in for a biopsy. A biopsy will be taken, a small piece of tissue will be taken and fixed in formalin and then embedded into paraffin. And from there, we will be able to extract DNA to investigate that tumor. It can also be extracted from the blood. In your blood, you have red blood cells that carry no DNA. It's one of the it's probably the only cell type that does not carry DNA. Oh, no, actually, there's platelets. But then there are white blood cells. Your white blood cells, your immune system that is present in the blood, they carry a copy of your germline DNA. There's also another type of DNA that you will find that it's normally diluted in the serum, so in the liquid part of the blood, and it's what we call cell-free circulating DNA. This is the result of a natural process that happens in your body, which is as cells die, they burst and their DNA gets spread into the bloodstream. Most of it gets removed and get degraded, but some of it lingers around. And this is true for your healthy cell, but is also true for your tumor cell in the case there is a tumor. And so you see that you have a number of places where you can get germline DNA or a mix of germline and tumor DNA. Okay, then once you have the DNA, there is a complex process. I'm not going to take you through all of them, but I just wanted to give you an idea of the time and complexity that it takes to get to this kind of results, where the DNA is basically taken out of the cell. It takes a couple of hours. It gets that sample gets then prepared. The DNA basically gets chopped. Some chemicals get added. It gets fed into a machine that will basically read it for 1 or 2 days. Then all this data needs to be crunched and reassembled and realigned to a reference genome. There is a whole process and a whole set of algorithms that I'm not going to cover today that goes and identifies which changes have been between the signal that I got from the machine and the signal and the reference genome that I have, and that helps me identify what we call mutations or changes. And that is a very computationally heavy process, which can take up to half a day and use a lot of CPU and GPU power. And then there is the actual interpretation of the results. And this is the part where we're going to go and dive a bit deeper into what it means to interpret those results. Now, as you see, this is a test that takes a couple of days to run. So it's not like a COVID test that might take 15 minutes to a couple of hours, or like an X-ray that basically the result is almost instantly available as you do that. And so it's a complex process, but it's also a costly process. Now, luckily, one of the things, I'll get back to the cost in a second. I forgot there's another part that I wanted to say. So one of the main activity that happens in this process is that once you have your biopsy, you do something that is called the genotyping or genotype profiling, where basically you are trying to record every single change that you identify in that sequence of DNA and attribute it to either the tumor or the germline. What happens is, if it's present in your germline material, most often than not, these changes will be present in either 50% of all the signals or in 100% of all the signals. If they are coming from a tumor, you will see them as episodical changes that are basically what we call point mutations or single read mutations. And that will basically tell you, okay, this is likely coming from the tumor. Now, this is a qualitative or semi-quantitative, if you will, kind of exercise when you do it on a biopsy. When you do it on the blood, you have an additional thing that you can do, which is you can repeat that exercise over time. And so one of the things that we have learned actually through this technology is that if I take a sample of your tumor before therapy, and if I take another sample of your tumor after therapy, the composition, let's say the amount of mutation that there are, the different type of mutation that exists in that tumor might have changed as a result of the therapy itself. It's a bit like when you treat with antibiotics and, if you have an infection, you give antibiotics and then the infection basically dies out. And so you can see the diminishing presence of that bacteria, but sometimes you might actually favor the emergence of a resistant bacteria species. Similar thing happened with oncology. Remember that these tumors are basically blobs of cells that are alive, and as all living creatures, they react to evolutionary pressure, and they do so in a very, very rapid manner because their metabolism is extremely altered. And so what happens is, as you go through chemotherapy, the composition of your tumor, which mutations are present and absent in your tumor, will change. And that change is reflective and informative of what is going on in the tumor itself. So this is another data point that is super important. As I was saying early on, the other thing is this is a very costly exercise, to the point that 20 years ago, to sequence a genome, it would cost around $100 million. So it was a very much of a research exercise. In the last 20 years, and especially, let's say, between 2007, 2008, and 2010, etc., in those 3 or 5 years, you see that the cost of sequencing has dropped in a way that is unprecedented. As a reference, there's Moore's law, that is a famous law in computing that says that your computing power doubles every, I don't remember if it's 14 or 16 months. So it's an exponential growth, and as you can see on the left, the y-axis is a logarithmic axis, and you see how the cost of sequencing has been decreasing faster than the cost of computing. And so we reach today where sequencing a human genome costs around $600. Now, it's still a very expensive test, but it's something that you start to be able to afford in a healthcare system. And so this is what is pushing also the adoption of this technology into the clinic, is the fact that we now have a cost-effective way to go and read the DNA from cells and get information about it. Okay, so all good, but as you know, not everything is always straightforward when it comes to science. So there are some major challenges when it comes to this. One of the first challenge is that the amount of variance that comes out from this exercise. So when these algorithms, when these techniques are used and these algorithms are run on the data that comes out from these instruments, you obtain a file. You will hear me refer to it many times throughout the rest of the presentation as a VCF file, a variant call file. And that file contains basically every single difference that there is between a reference genome that you see called here as a ref, and what is the actual identified letter, and it tells you in which chromosome and at which position. So, and these algorithms, they list all the possible sequence that do not match the reference and their coordinates. So what happens is that after you've sequenced a sample, you might end up with something between 500 to 4 million variants. And so this is what I said early on. It's just unimaginable for someone to go through a list of 4 million variants and being able to even guess which one might be relevant, which might be just a difference in your eye color, or the fact that you're better at metabolizing caffeine and so forth. So this is the first and one of the major challenges that happen here. Luckily, we have devised some clever strategy, such as filtering based on previously known sequence and so forth. We're not going to go too much into that, but there are some clever ways to reduce this number of variants. The second challenge is that you have basically just the position and the letters. Now, if you go and look in any guidelines, in any literature, in any clinical trial, very often things will be named in a different manner. So the other problem is it's the conversion of this reference position alternative into a language that is understandable to humans. And so, unfortunately, over time, there have been multiple standards that evolved. And so you see here, I tried to color it with different colors, how a mutation might be called in very different manner, although it's trying to say pretty much the same things. Also, this translation to human readable language, right? EGFR L858R, that basically makes it much more easy for humans to be able to interpret them. But as I said, there are different standards, and so you need to start to kind of make some inference around what standard you're going to pick, which one are the most appropriate for the type of use that you need to do with that information. And as you see, also what I mentioned early on, you might identify variants that basically have no clinical importance for that particular disease, right? So the fact that that particular variants encode for the color of your eyes does not tell me anything about whether a drug will be effective on this particular type of lung cancer or not. Just like if I have identified a variant that it's very common in breast cancer, but my problem is that there are development challenges in the patient, that does not necessarily make it relevant. So we start to introduce the concept that the relevance of a particular mutation is dependent on the condition that we are trying to investigate. Okay, the last big challenge, I would say one of the last big challenges, is the fact that the amount of information that goes into the decision process of what you're going to do once you know all of this. Are you going to do treatment, or are you going to just keep monitoring? Are you going to go medical or surgical treatment? Are you going to enroll the patient in a clinical trial? Are you going to give an approved drug, or are you going to use an existing drug that is maybe approved for something else, but because there is a similar mutation, you're going to use it off-label, and so forth. So there's a lot of potential decision that the physician might take, and in doing so, they need to consider all these data points that you see from here. And I know that some of the people on the audience are actually very much in the clinic, so you know that you have to look at your, let's say, the general clinical data of your patient. You might look at the anatomic pathology result, the lab result, the result from sequencing. You might look at what has been now approved and what is in the guidelines. You will look at what is being recently published or what you've seen recently at some conferences. You look at real world data. And so all of this needs to be fed into this decision system. And again, this becomes very, very challenging to do for a human. So on top of that, there's another layer of complexity, which is all of these things are not static. So I took this slide actually from a very old presentation that you see, how many approvals or how many abstract or publications there are in 2016, 17, and 18. So we're talking about 5 years ago. And every day, every year, these things get updated. New drugs are approved. New clinical trials are initiated. New publications come out. New medical guidelines are released, or old medical guidelines get updated. So all this stream of data, it's not static, it's dynamic. And so one of the key challenges is how do you stay on top of all of this while making the interpretation on your specific case. So the current approach that we have, and I'm going to mention just one of the products that we have. I take it as an example. This similar way of thinking, it's applicable to many other products that are out in the market. I just copy and pasted the slide because I have it. So don't think, please don't take this as a promotional thing. It's really just to make you understand the concept. And also, I'm going to focus on the interpretation of variant in the context of oncology, but a very, very similar approach might be taken in the interpretation of other diagnostic data in different pathologies. And so when we talk about clinical decision support, we're referring to a broad class of tools that basically helps diagnosticians and clinicians to make decision about what is the appropriate course of action based on the data that you have. So what we have for the specific use case of oncology, we have a tool called Navify Mutation Profiler that takes these VCF files and compare it with a knowledge base through a set of matching rules, and then spits out something that it's easy to digest for a pathologist or an oncologist. where basically identified what are the relevant variants that have been found, what are the therapies that are associated with that, what are the clinical trials that might be relevant to that particular thing, and do that through what we call a user interface that helps with interpretation. Now, I want to focus especially on this left part, on this molecular knowledge base. What do we mean by that? Well, what that boils down to is basically it's a way to take all this information from clinicaltrials.gov, PubMed, all the different oncology association that release guidelines, the FDA, all the different food and drug regulating authorities, and structure them in a database in a way that can be then used in an automated or semi-automated way. Then we also ingest existing genomic database that contains information about which variant might be pathogenic or non-pathogenic, likely pathogenic, and so forth. So some sort of way of filtering and standardize the annotation. As I mentioned early on, one of the key challenges that we have here is naming standardization. We standardize all of that, we merge this content, and then we feed it to a product, a software product, in which a customer, or let's say a lab, can import their VCF. This VCF will be compared to some sort of rules. If you are coming from the informatic world, it's basically like a giant join between my file and this giant table in the database to figure out what is actually relevant out of that knowledge base that contains all the known knowledge up to date on the oncology space, what is relevant for my particular case. And as you can see, one of the key challenges really is really keeping this knowledge base up to date. And you will see here one particular thing that is mentioned is this NLP automation. There's also some manual curation. This is by no means a fully automated process. We are not yet there. But this is exactly where NLP, or natural language processing, comes and help. Okay, so what is the role of NLP? I'll go and do a little step back for those that are not familiar with NLP. I'll spend 2 minutes introducing a couple of key concepts that are useful to know. The first part about NLP is basically how do we turn text, which is something that humans understand well, into something that computers understand well. And what computers understand better is, generally speaking, numbers. So one of the first and most basic approaches to NLP is simply to turn text into numbers. How do you do that? The most intuitive and, let's say, the very earlier approach to NLP is simply to count the words. So if I take the Wikipedia page about HIV and I count each and every word and I go through the document and count how many times each word appears, you will see that even just by doing a simple word count, you can figure out pretty quickly that this document, this piece of text, is about HIV and some sort of virus and viral particles, cells, etc. So you see how just a simple word count can turn can extract some useful information from a piece of text. Now, there are more sophisticated ways. There's way to normalize for things that we don't care about. But this is, let's say, one of the most basic form of natural language processing. Now, once you have things in number, you start to also be able to do very interesting things. For example, I can count how many times the word HIV happens in a particular document. Or I can take a bunch of documents and say, okay, this other document, the word that happens a lot, it's cancer, but it doesn't contain anything about HIV. So you can imagine plotting them on this 2D plot. You might find a third document that contains about, let's say, a high number of word count for HIV and a high word count for cancer. And that basically means that most likely that document is about HIV and cancer, and most likely about their association. Now, if you repeat that You also might find documents that have none of them. And once you do over and over with various pieces of text, you can start to see that some cluster might form. And what you can do then, it's basically make some inference and say, hey, all these documents are part of a cluster. And that cluster, if you take the center of that cluster and plot basically what are the keywords, tells you what the topic is in that particular cluster. This is a very useful transformation when you start to tag documents, when you start to add, let's say if you are ingesting, I don't know, 20,000 publications, and you start to simply want to tag which one are the ones that are about HIV and cancer that I want to further analyze, well, those in the cluster 2 are your first screening criteria. Of course, there are more sophisticated way of doing it, but this is kind of a very, very crude way that kind of leads to very useful result with very little effort. Now, I made this example in a 2D fashion, right? Just taking HIV and cancer. But the good thing about compute is that it can scale to an infinite number of dimensions, right? For us, it's very hard to think over 3 dimensions. But for a computer, there's no problem in assigning a document into a space that has as many dimensions as the total number of words that exist. And so you can start to imagine this transformation of a document to a space where that document lives as a function of, let's say, how many words of each type it contains and where does that place you in this n-dimensional space. And that allows you to basically do this clustering and this kind of like topic inference on a very, very large scale and across basically in a very unbiased way without knowing a priori what you're trying to get out of these documents. Okay, this is one of the first approaches that has been taken in NLP. Another very important concept to understand, and that you will see why it's important, is the concept of how words relate to one another. So what I can do is I can create a matrix where I say, okay, I count how many times the term number 1 exists in document 1, 2, 3, and so forth, and I create a simple matrix. With this matrix, what I can do is I can also look at the correlation between terms or the correlation between documents for that matter, but let's take the one between terms, and I can start to build graphs. Graphs in which each node represents a particular word and each edge represents the strength of the connection between those two words. How often do they appear together? Are they often together? The edge will have a higher value. If they are less often together, the edge will have a lower value. And you can start to apply this, and you start to build what we call as domain knowledge graphs. This idea of mapping out a domain and represent it in a graph form that can be then expanded and explored by computational methods. Okay. Okay, that's what I was saying. Term 1, term 2, basically those are your nodes, and then the edge represents the strength of that connection. Okay, now, once you have all of this, this is when things start to kind of become a bit more real. So at this point, what you can do is to take a lot of, retrieve a lot of text from literature, PubMed, publishers, etc. And you start to do a set of transformations based on this concept that I mentioned early on, such as recognizing variants, gene, diseases, drugs, variant groups, cell lines. Then you can start to normalize those concepts, sometimes using, for example, the fact that they cluster all together. You can also start to associate concepts with one another based on particular relationship in the text. And you can start to build structure in your data. Okay, that's clear. Now, what do I mean by recognizing entities in the text? I brought here an example. You might find a piece of text that says E1 gene expression induced susceptibility to killing by NK cells, etc., etc. And you see how after some of this after this processing, you start to have things highlighted that represent particular concepts that are now standardized, queryable, and allows you to make educated guesses about whether that gene that is in that particular DNA region has some sort of interaction with this type of cell or this type of viruses, what proteins are involved, etc. So you see that you can start to take this, let's say, unstructured text and turn it into something that has a structure, and that structure can be used to make guesses and educated predictions. What you can do is also use computational techniques that are based, for example, on graph theory, on exploring the network and searching for relationships. So there's a whole field of graph knowledge graph and knowledge exploration and graph theory that you can use to retrieve and to rank relevance of nodes. If you remember in this structure that we highlighted early on, where each term it's a node, you can start to explore nodes for adjacency, for relevance of that particular term to with respect to one another, and so forth. And you can also build those graphs in a way that span multiple type of networks. So you can have, for example, a network that represents protein-to-protein interaction, a network that represents a gene pathway, and a network that represents a disease. And you start to connect those networks and to build a mesh that spans this heterogeneous network that connects genes with diseases, with protein interaction, and you start to use these networks and the network that we built from the text and start to explore how those are connected. And basically you can get things like co-expression, co-regulation, whether they co-occur in the literature, whether there's a gene to disease association. And once you have that, you can start to rank those things in a way that it's informative and it tells you something about this particular set of genes or these mutations in the context of this disease are likely to have this effect. Okay, so this is really kind of like where it boils down. This is where all what we have learned throughout today about the NLP part, about the genetic part, about the knowledge graphs and etc. kind of come together, and you really start to make those inferences using computational methods. In this case, we're just using logistic regression models for prioritization, but there are AI based models. There are many other ways you can get to that. But basically, this allows a human to prioritize the important thing in the context of this disease, in the context of what is new, etc., while interpreting the result that come from the sequencing. Okay, last few words looking ahead. So up until now, I focused very much on what we call traditional natural language processing, or let's say NLP, which really has been, let's say, the bread and butter for the last decades. But in the last few years, a new class of NLP technique have emerged, which is everybody knew it's famous. It's aware of this particular class, which is large language model, ChatGPT, BERT, and so forth. So this idea of using new machine learning and deep learning methods and transformers, etc., to extract information for text has a lot of promises in what can be done. And if I look at looking back at what I was sharing early on how we do it in terms of the building of the knowledge base, there's two major areas where these techniques, we expect them to have a major impact. The first excuse me, the first one, it's really on the extraction of the scientific knowledge. So here really going in each of these elements that we mentioned early on, entity recognition, concept normalization, concept association and mining, evidence ranking, etc. These are all based on other type of algorithms, and there is a potential to be replaced, to replace some of them or all of them with LLM-based algos and see whether we can have better performance in terms of accuracy, in terms of speed, scales, etc. But also in terms of complexity. However, the other part, which is also very, very interesting, is the potential for LLMs to start to help or even in the future take over some of the review activities that the humans are currently doing. Because at the moment, these NLP pipelines, they just create this knowledge, they consolidate it, and then a human will review it and will validate it. And maybe we can go to having 3 humans to 1 model and 2 humans, or 2 models and 1 human in the loop, trying to get to this review of what those pipelines have generated. So these are currently areas that are under exploration, at least internally, but also in general in the research space. There's a lot of work being done on LLMs for entity recognition, for concept association, etc. How do you traverse the embedded space to get concept association and so forth? So these are really areas where these models are poised to change the way we do NLP, but the goal, it's still the same. The goal is how do we get all this unstructured data here on the left in some sort of structured form that allow us then to effectively use it when it comes to interpreting the result. And as usual, I leave you with what it's something that is always very important to remember in our field, which is algorithms are not the ultimate solution for everything. Biology is complex, healthcare is complex, we are complex, humans are complex, and so they can help us reduce some of that complexity, but we won't get rid of all of it. So that's it from my side.
Thank you very much. Thank you very much.